I mean, I'm going to talk for a little bit, but I was going to introduce you formally. Awesome. We'll do that. All right. Welcome, GRIN2B families, to the 2026 GRIN2B Foundation Family and Research Weekend. My name is Phil Ash, and I'm on the board of directors for GRIN2B Foundation. I'll also be an MC of sorts for a lot of this weekend, so for better or for worse, you'll be seeing and hearing me. It is an honor to be with all of you. We're so excited to see you guys. Really quickly, show of hands, how many of you are attending a conference for the first time? All right, very cool. All right, how many of you have been to 2 GRIN2B conferences? Okay. Do I have 3? 4? Okay, because we only had 4. That 3, that's pretty impressive. That's awesome. All right, let's try this. How many of you are newly diagnosed families within the past year or 2? Okay, awesome. To our newly diagnosed families here, online, in our entire community, welcome to our small and fierce GRIN2B Hive, right? And let's be honest, if we're being honest with ourselves, this is a very exclusive club. It's the club that before your child or family's member's diagnosis, none of you knew about it. And if we'd told you about it, probably none of you would want to be in it. But here we are. But in all seriousness, speaking for myself, I can say that after 3 other family conferences, 10 years of GRIN2B Foundation, and 12 years since my daughter Lucy's diagnosis, that this is a club that I don't think I could do without. This exclusive community has changed my life for the better. It's made me a better person, and I truly hope that it's doing the same for all of you. I'm going to speak on behalf of our board of directors that we have all done our absolute best to give you all what we hope will be an unforgettable weekend of learning, discussion, and really most importantly, togetherness. To everyone attending, but particularly those who are attending a GRIN2B conference for the first time, this will probably be a very personal experience for you. You can partake in as much or as little of this as you like. Some of you are going to absolutely love all of the science and research discussions. Some of you are going to love the family panels, the group discussions. I know that some of you are going to get overwhelmed at some point by something between now and midday Sunday. And all of that I just said is completely okay. This is not a college course. There's no quiz. There is no right or wrong way to do this conference. If you need to unplug and step away, please do so. Take in as much or as little as you like. Every single session is going to be live streamed or recorded and uploaded to our YouTube channel, so you can always watch anything that you missed. But above all, what I really hope everybody does this weekend is connect with one another. Every single one of us in this room are affected in some way by GRIN2B-related neurodevelopmental disorder or a GRI disorder. Literally down the hall is a group of some of the most intelligent scientists, researchers, and people that I have ever known, and they are all here for us, researching and collaborating in the hopes that they can make our lives and the lives of our diagnosed loved ones just a little bit better. Perhaps most importantly, we are in a room of people who understand completely what life is like for you and your family. You don't have to put on a brave face here. You don't have to wear an everything is okay mask, because all of us experience the joys and the triumphs and the small victories, but we also face the setbacks and the heartaches, the stress, the fears, and the never-ending roller coaster of living with rare disease. So let's all try to be here this weekend for each other. So one more time, welcome to our family and research conference, and thank you for your attendance and support of GRIN2B Foundation. All right. I do have a few announcements that I want to make before we really get into meat and potatoes, and I'm going to introduce a very special person here in just a moment. First of all, I want to thank our incredible sponsors that are helping to make this weekend happen: GRIN Therapeutics, ARIA, Aligning Research to Impact Autism, Ludbeck and SFARI, Simon's Foundation, Praxis, Cure Grin, Ovid Therapeutics, myself and my wife, Liz Marfia Ash, and the Metric family as well. So thank you very much for all of our sponsors for helping us. And please make sure during breaks that you visit the tables. I know a number of you already have. Please do so. They're here for you to help answer questions. And yes, welcome to our Hive. In just a minute, I'm going to introduce our executive director, but I have a couple of other things I want to say first. So first of all, if you do have a question, and I believe there is a QR code in your folder that you received upon registration, so don't scramble to take a picture of this unless you already have a question, in which case, awesome. But if you do have a question, you can scan that QR code, you can type that question in. We will be looking at those questions periodically throughout the weekend, and as we have time to, and some of our Q&As that we devoted for each day, we'll try to get to some of those questions and get those answered for you. Very important announcement, we have blood draws that are happening today in the Heart Room, which is about kitty corner down that way. And so I do want to say that if that's something you're planning to do, to try to make that a priority, the company that is doing the blood draws, they have a hard out at 4 p.m. So, we want to make sure that if that's something that you want to do, that you get that in, so that there's not some long line that people are waiting on and things like that. So please make sure that you are taking advantage of that. I think we have an open slot currently if someone wants to get that done, because who doesn't want to get poked, right? In the arm. Our BioGene study, which is happening right here in this hotel, there are slots still available. So if you would like to participate in that BioGene study, you can scan that QR code and make yourself an appointment. Also, I don't have a slide for this, but right outside the hall and to the right, there is a table. It's for the Colorado GRIN variant registry. Please sign up if you haven't done so during one of our breaks. Again, just getting your information out into the hands of people that study it is so important when we're talking about an ultra-rare disorder such as GRIN2B. And with that, I think that's all I have. Yes, I'm going to introduce our executive director for GRIN2B Foundation. Everybody give it up for Alyssa Taylor-Bastillo.
Thanks so much, Phil. So first and foremost, it is such a pleasure to see each and every one of you today. I do not have remarks prepared as Phil did. I tend to be a person who goes off of the cuff, but I very well know this difficult path that most of you, well, all of you in this room have gone through with getting a GRIN2B diagnosis for your kids. My child does not have GRIN2B. My middle son has a completely different rare disease, ultra rare. So I also know how it is to navigate through such a difficult time, and I commend each and every one of you in this room for dealing with the adversities and for coming out to join us this weekend. So welcome, and thank you for being a part of this impactful program. So we're going to get started with just a couple more reminders. We have our 7th annual Be Active for GRIN2B fundraiser right around the corner. It will be our program for our foundation. It is how we raise funds to bring programs like Family and Research Weekend to you guys, but also some of our support programs where we're able to support families who may need some additional items that are not covered by insurance, and some other necessities for their day-to-day care. So if you're able to, you can scan the QR code here on this page to start your Raisley website and get started. We have tons of materials that you guys can reference to for help. Never feel that you are alone at any part of this journey. We're always here to answer any questions you may have or provide any resources. Just a quick reminder, and just to reiterate for this as well, This is also how, I know Phil mentioned that we have a ton of researchers down the hallway. A lot of the funds that we get to be able to support research initiatives that help our community as a whole is through fundraising. We actually have a research opportunity that we're looking to fund this year. So this year's Be Active for GRIN2B is incredibly important so that we raise enough funds to be able to launch and get that research started. Just a reminder for those who are have their children signed up for childcare, Jovi, our partners for nannies and sitters, they will be here tomorrow and Sunday. They will be in the Chesapeake and Annapolis room starting at 7:30 tomorrow up until 4:30. So you can go ahead and drop your kids off, I believe, starting at 7 a.m. Drop off those forms that were emailed to you that have your information, your child's information, and any medical needs that they may have. And they will get you guys all signed in. For meals, you would go and pick up your kids from the child care room, have meals with them, and then Jovi will take them back for care for the time in between. And then Sunday, they're back over there from 7:30 to 11 a.m. We also have a changing room, an actual changing room this year for those of you who may need it. It's across the hallway in Dirksen. There's a partition in there for privacy, so you guys can take your kids in there to change. We also have an actual changing table in there this year just to make sure everyone has what they need. So go and check it out if you need that amenity. It's right across the hallway. And then last but not least, we will be doing an outing to the Smithsonian National Zoo on Sunday. So I hope that you guys are all able to come with us. The weather should be breaking overnight from Saturday into Sunday, so it's going to be much cooler than it has been the last 2 days. I'm local here in Maryland, so I'm welcoming you guys to my city and also apologizing for the air quality and the horrible temperatures that we're having. So rest assured, Sunday should be a much better day for an outing. We will meet at the ticket counter at the Smithsonian Zoo adjacent to the parking lot at 1:30. So meet us there as a group. If you have any questions, feel free. My business card is in all of your folders. Send me a text message or give me a call if you can't find us, and we will find you. We'll figure out a way to get connected with you. And if anyone needs a ride or transportation to the zoo, please feel free to send an email and let us know. We have some families who have space in their vehicles for carpooling. So if you need a ride and don't want to Uber, let us know. We'll try to get you paired up with someone who has availability. And I think that's all that we have. So thank you guys again, and we hope that you have an amazing time this weekend, full of connection and learning, and we're excited to get things started. So thank you so much.
Thanks, Alyssa. Okay, and on that note, one of the things that we really wanted to do for this conference is to make sure we have a lot of time for you guys to just kind of talk and be with one another. And so, but we also wanted to maybe structure that a little bit too. So that's kind of what we're going to do to start off with our Hive Mind chats. And so I'm a high school teacher, if you don't know. And so this is sort of very much like an activity that I would have some students do, but I actually think it's really powerful, and I think it'll really lead to some really great discussions. So if everybody takes their ID, flip it over and look it on the back. So on the back, you should see that you have 4 colors, okay? I want you to read them from, if you're looking at it this way, I want you to read it from left to right, okay? So, for example, if your first color is blue, the blue table is there back by Kelsey. Kelsey, raise your hand, okay? If it is red, it's over there. I think that's, there's a large Post-it note laying there. That's the red table, I believe, right there. It must have gotten moved. And then there is a yellow. There's also placards that have them there. So essentially what we've done is we've tried to randomize you all, okay? So you're gonna go to those tables, and on those Post-its, there is a question that I just would love for you guys to discuss and talk about, and if you would be so willing to write responses on those Post-it notes. Any of the white space is fine. We don't care about your handwriting. We just want to see what you guys, what's working and what's not working, and what's something that's going well and something maybe that you're struggling with. These are all questions really kind of about day-to-day life and sort of what it's like with your kiddos and your families and things like that. And so we're going to give a little bit of time. So in theory, about 15 minutes per table, and then we're going to switch, right? So essentially what we'll do is we'll have timekeepers and say, and after, so for example, if your first color is red, then I'm going to say, okay, you need to move to the next color to the right on your placard. So maybe that, and then you go to that next table, and then you go to the next question and continue. Does that make sense? It sounded great in my head when we planned this a couple of months ago. So, all right, cool. So I'm going to ask then, if you guys are able to, there should also be a pile of pens and Sharpies by the Post-it, so you shouldn't have to necessarily bring that stuff. And if you want to leave your stuff at the table you're currently at, that's fine. We won't steal anything. But go ahead and move to your first designated color spot and start getting to know some people and start seeing if you can give us some insight on what's happening in the day-to-day. up the conversations, but I'm just going to ask you to have more conversations. So what I want you guys to do, flip your ID badge back over. Again, looking from left to right, you're now going to move to your second color table. In theory, everybody should be in a pretty much different group, okay? Different group, different question set. Let's go ahead and move. We're going to take 15 more minutes, okay? 3 more presentations before we conclude for Friday. Just a couple of announcements before we introduce our first speaker. So blood draws are happening until 4 o'clock sharp. I believe it's 3 o'clock now, so you have 1 more hour if you wanted to try to get a blood draw in, and that's in the heart room, right across the hall and down a little ways toward registration. They might be able to take you in if that's something you'd like to get done. And then also above the water station there, and then also in Salon 3 above the coffee station is a QR code for the BioGene study. So again, if you would like to sign up and have that done, it's an EEG. They are here and equipped for that. I think it's room 1035 or 38. I don't know. If you fill out the form, they'll let you know what room it is. So you're not, knocking on somebody else's door. But that's what, 1035? Yeah. So you'll be able to get that done as well. If you want to scan the QR code, you can get that taken care of. Okay. So welcome back, and we are going to start our presentations this afternoon with Dr. Kristen Park. She is the director of the GRI Center of Excellence at Children's Hospital in Colorado. She's incredibly awesome in all that she does to help support not just this community, but GRI disorders in general. And she is going to give a presentation on a beginner's guide to GRIN2B. So please welcome Dr. Kristen Park.
All right, thanks. Let me see how to, is there a pointer maybe? Doesn't, it does say that there's my name. Let's see. Okay, here we go. So this is what my title was, the alternative title, GRIN2B 101. So this is going to be the basics. Hopefully they'll be the basics, and we'll see how it goes. All right, so what we're going to cover today is a little bit about what happens with neurotransmission in the brain, how is the NMDA receptor involved in that, what are the basics of how to interpret genetic variations and interpret genetic test reports, and then finally, what are some of the clinical features that we see with kids who have a GRIN2B NDD diagnosis. Okay, so this is kind of an overview of how the brain kind of works. I think of it as that the brain communicates in primarily 2 ways. One way is with electricity, and the other way is with chemicals. And so those 2 things kind of work together to send messages from neurons to other neurons. And it also kind of has 2 ways to send those signals. One is a gas pedal that says go, that excites and makes things happen in the neurons. And the other is kind of the brake that says stop and quiets things down and makes things calmer. And the excitatory neurotransmitter in the brain is going to be glutamate. That's where we get our G for GRIN, is from glutamate. And then, well, unfortunately, I guess the inhibitory also has a G for GABA, but it stands for different things. And most of the synapses within our brain are actually glutamate synapses. 95% of that chemical transmission occurs with glutamate. Glutamate has different types of transmission. It sends a very fast signal, which is the ionotropic signal, meaning that the signal gets translated into electrical signals, which is what ions are, right? Charged particles like calcium or potassium or sodium or magnesium that flow in and out of the cells to help send that message. And that is very quick. That's milliseconds that's happening in most of the neurons. Whereas the metabotropic side of things is a lot slower, and we usually don't talk a whole lot about that in this group because the I stands for ionotropic, and so that's where we get the I in GRIN, right? So the metabotropic are things that respond to kind of stress or hormones or, sickness within the body. Those changes take place on a more like a week to, several weeks to month timeline rather than a millisecond timeline. And then the ionotropic receptors themselves are divided into 4 subtypes. And those subtypes occur based on what we have discovered in the lab binds most strongly to the receptors. So we have NMDA, AMPA, kainate, and then delta. I think that was because it didn't necessarily behave the same as those other 3 channels. And I'm not going to tell you what they stand for because no one's going to remember it. Okay, so you may have also seen this picture in some other talks and slides. And so this is kind of like a number of different ways to represent this category of ionotropic glutamate receptors. And so you can see those subtypes are listed there on the top in the different colors. And each of those kind of families of receptors has a number of different constituent parts. So under AMPA, you can see that it has GLUA 1, 2, 3, 4, and those are kind of different specific types of subunits to those receptors. And the NMDA receptors have kind of a number of different boxes there because we have GRIN1, which is, and the reason that these letters look different is because this is the name of the gene that codes for the protein. So GRIN is actually GLUN, and then you put the number after it. So GLUN1 codes for GRIN1, whereas GLUN2A codes for GRIN2A. So it has different, again, different families, 1, 2, and 3, and then there are some subunits as well. And then at the bottom, you can see the NMDA receptors in general. They combine into what's called a heteromer or a tetramer, meaning that it has 4 subunits to make up one receptor. And the GRIN2B or the GRIN receptors are often heteromers, which means that not all the subunits are the same. So for a GRIN2B receptor, it's usually 2 GRIN1 units and then a combination of other GRIN2 subunits to make the 4 components of the receptor. That is not the same for all the receptors in the ionotropic glutamate family. You can see that there's different combinations. The kainate receptors can have homomers, meaning all the subunits are the same. But for GRIN2B, the combination is always 2 GRIN1 units and then a combination of other GRIN2 units. Sometimes I think GRIN3 might go in there too, but I don't really know a lot about GRIN3, I'm going to be honest. So that's kind of that left-hand part, and then you see those different colors. They don't show up that well, but the glutamate is a little bit like an ivory color, and the glycine serine is more white. That's because these receptors have different things to which they bind, and they bind more tightly to some than to others. And then on the next, you'll see that stretched out portion. That is kind of like a pictograph of what the gene looks like and its various different domains. So from the left in purple is the N-terminal domain. Then there's S1, segment 1, M2, M1, M2, M3, S2, and then there's M4, and then the last part is the C-terminal domain. The C-terminal domain kind of hangs out inside of the cell. And those different areas have different purposes within the gene. Some place might be someplace where something binds, another place transfers along the membrane, and that's what that picture underneath is showing, those different colors translated into kind of what the gene does or what the protein does. And so you can see there the orange area is called the ABD, the agonist binding domain. So things come and sit in there, bind to the protein, tell it what to do. The TMD stands for the transmembrane domain, and those are those different numbers. They span that and help to anchor that receptor in the cell membrane so that it can receive those signals and then open and let ions through. And then this last part over here on the right is what's called a ribbon diagram. And that's the scientists kind of taking the actual sequence of DNA and turning it into a picture of what the coiled and folded protein looks like. And we can use that to plot where different variants occur. Okay. So, this is, again, a little bit of a different way to view that same information. So we talked that the brain transmits chemically and electrically, and that happens in these little teeny spaces in between two nerve cells called synapses. And a synapse, and to transmit those signals, we demand a lot of that synapse, and we demand a lot of that communication. We need it to be efficient, and we need it to strengthen when we learn something so that we can remember it next time, and we need it to change over time. We want to be able to forget our old phone number and learn our new phone number, right? So we need to let go of that old reinforced connection and make a new connection as we get a new phone number. Again, glutamate is the trigger for this signal, and it is released in a little package from the sending neuron and then goes across that little space and binds to the receiving neuron. And the receiving neuron needs to already be active, which implicates AMPA. Remember, AMPA is a different type of glutamate receptor. That's the GRIA receptors. And then it has something called a magnesium block. So in order for the channel, the NMDA receptor, to be active, it needs to already be kind of going in that direction, and the magnesium needs to be out of the way. That allows calcium to flow into the receiving neuron and send the message on to the next neuron. And it does act in a little bit of a cycle, because part of the message that gets sent on is to strengthen that signal by adding more of those AMPA receptors so that the next time the signal gets faster. And the way that we say that kind of colloquially, and what we learned, kind of in medical school, is that neurons that fire together wire together. So when you get synchronous activity and neurons that continually trigger each other, that makes the signal stronger the next time. And the scientific word for that is called long-term potentiation. And you might hear that because that's one of the key components of learning and memory that's studied on a cellular level by academic scientists, in little dishes and things. Okay, so the next point to say is that, again, we're talking about our phone number, we're talking about learning and memory. Not all the signals that we get need to be strengthened. We don't want to learn everything. We don't want everything to get stuck in our brain and make some sort of connection. So the NMDA receptor acts like a quality control to say, hey, this happened, and this was something that should be important to remember. And so we should strengthen this synapse. The other thing that they do is that when our brains are growing, the cells that form our brain, our neurons, have to start in the center and migrate and form connections. And glutamate acts like a guidance system, telling us how to do that, how the neurons to do that, and how to form those connections. And that's because the calcium that the glutamate receptor lets in acts as a signal for kind of the scaffold that helps those neurons to move into their destination. And so I think this is important because some kids with GRIN2B and other GRIN disorders can have malformations in how their brain forms. And it's probably because of these signals that are happening and this scaffolding that is not organizing itself in the right way. Again, we talked about development and learning. That's a process of strengthening these useful connections and weakening connections that are not active. And again, if we had long-term potentiation as the scientific term for learning, long-term depression is the term for weakening those synapses. It's also called pruning, if you hear that word. So the other thing I think that's important about GRIN2B to understand is that, say, while GRIN1, we looked at those heteromers, right, those 4 components of the NMDA receptor channel, GRIN1 is always present in those and those groups. But GRIN2B is not always present. Other two proteins will come into that channel. And the order and the space in the brain in which that happens is kind of pre-programmed. And so, in places that are very active while the baby is developing, maybe they need a little bit more 2A, and then a little bit later they need a little bit more 2B, and then another section of the brain needs a little bit more 2D and its receptors. So there's a very specific profile that is used for GRIN2B that happens as a baby is developing both in utero and afterwards. And I think that's important because that helps us, or that makes us have to take note as scientists of when is an intervention happening? When are we testing something? Is it when GRIN2B is really active or when it's kind of less active in development? And then the last thing I would say is that glutamate, again, because it's at 95% of our synapses, we've talked a little bit about learning and memory, we've talked a little bit about migration, but glutamate is involved in a number of different neuronal processes. It's involved in movement, it's involved in our sensory system, it's involved in pain. It's been known to be around blood vessels that go to our brain, and so dilation and contraction of those blood vessels, and then autonomic regulation, which I think we talk a lot about in clinical symptoms. So glutamate is a very important signaling, protein or compound in those processes. Okay, so that was a lot of science. Now we're going to move into genetics, which is also a lot of science, but we'll see how it goes. Okay. This is science that I like a little bit better, so maybe it'll be a little easier. All right, so this is kind of the what is called kind of the central dogma of genetics, right? That inside of our cell, inside of the nucleus of our cell, we have chromosomes, and in each of those chromosomes are genes that are all coiled up together, and you can unravel those coils and read the sequence of letters in order to form a protein. And a protein is something in our body that will go out and do something, like become an NMDA receptor, right? And so the process that we're going to look at, each of us has 23 pairs of chromosomes, right? We get one from our mom, one from our dad, and we have 23 pairs. That makes 46 plus our sex chromosomes, right? An XX for a girl and an XY for a boy. And then we have 20,000 genes total, many of them on various different chromosomes. All right, so these are some terms. We're just going to have a little glossary here. So a gene is a segment of DNA that codes for a particular protein. Again, we're going to talk about the GRIN2B gene, which codes for the GRIN2B protein, okay? And then a protein, again, is a molecule in our brain that has some sort of role in the body. Some of them, provide support and structure, like our collagen or our bones. Others of them send signals, like the GRIN2B protein. We have our immune system, which is formed of proteins, and proteins also are responsible for carrying out chemical reactions, like turning our food into energy. So proteins are made up of small units called amino acids, and there are 20 different amino acids. They are not the same. They have different properties. Some of them like to be surrounded by fat. Some of them are big. Some of them are small. Some of them are positively charged. Some of them are negatively charged. They have different shapes. They bind with each other differently. So each of those things is like a word, or in this case, as you see where I'm going with the pictures, an ingredient for the recipe of our cake. And each of those amino acids with its different properties is coded for by 3 letters. And that is called, we'll call it a word here, but in general it's called a codon. And the sequence of those amino acids kind of determines how the protein gets formed. Once that sequence is strung together, it has another kind of process that it goes through where it kind of folds itself into its ultimate shape, that little ribbon diagram that we saw that makes the channel that allows the calcium to come in. So it's not only the sequence of the things that gets strung together, it's how that sequence then folds itself into that channel. All right, so how do we do that? How do we turn a gene into a protein? I asked AI to make this picture for me. So you start with your DNA, right? It's a double strand, the double helix that we've heard about. That DNA gets separated and read, and it's copied into inside the nucleus. The next step is called splicing. And I think that's important to know. So splicing, the boxcars of the train. It keeps the train together, it does something important, but in the final product, that's not what's going to get taken out and passed on to something else. And that's called an intron, and it stands for intragenic or intervening regions. So this mRNA signal, before it gets out of the nucleus, they kind of go through and cut out all of those little linkers and smush all of the exons together so that that's what's being read at the next step. And the next step is that it goes to something called the ribosome, and then another kind of RNA called transfer RNA brings in those different amino acids that are represented by those little circles and strings them into the sequence of the protein, and that's called translation. And then finally, once all of those little strings are put together, the chain of those amino acids folds into the final form of the protein. Okay, and I think this is an important point to understand because this has some bearing on what kind of testing is done for people. So if your family member gets a diagnosis from whole exome sequencing, that means they only looked at those boxcars, and that's where the variant was found, versus whole genome sequencing, which looks at the boxcars and the linkers. The linkers are probably 85, they're like 90% of our DNA, but they don't contain as much information, at least not as we understand it. Most of what we understand about genetic disease is located in the exons. So usually whole exome sequencing is sufficient to identify genetic variants. Just for reference, the GRIN2B gene contains 13 exons, 1,500 base pairs, which are the letters. And then if we again divide that by 3, that means we have about 500 amino acids within the GRIN2B gene. Okay. So now we've understood how we get genes and proteins. Now we should talk about how we get variations in genes and proteins. And so a variation is any alteration in the sequence of that DNA that is different than what's seen in a large majority of people. And those variants can have different significance. So sometimes those variants exist in the general population because we are all different. There are probably about 3 million differences between anybody's DNA in this room that's not related. And that's because we need to evolve, right? We need to grow and adapt to different environments. We need to be resistant to different diseases, and we're all slightly different from each other. So some of those variants, though, can occur in places that are important for how our body functions. And so those would be what we would call pathogenic variants, meaning that those variants occur someplace that's important for the function of that gene, and if you have a variant in that location, it can cause you medical symptoms. There are some that we don't really understand, and those would be variants of uncertain significance. And that means, we haven't seen this in anybody, but we're not certain that it causes medical symptoms. We just really don't know what the answer is. And sometimes people will use the words for disease-causing variants and refer to them as mutations, but that's not kind of quite as common. Okay, so now we're going to talk about what types of mutations or variants occur in these genes that cause symptoms. So the first type is a missense variant. That's a change in one of those letters that results in typically the substitution of one word for a different word, one codon for a different codon, one amino acid for a different amino acid. Sometimes it can be silent. There is redundancy in the code. We talked about there's 20 amino acids, 3 letters, and you can combine that and you get some extra. And so it is possible to change one of those letters and not actually change the amino acid that's positioned there. And most of the time that doesn't cause any problems. The next is something called a nonsense mutation. So that is kind of a stop sign to the body. Every gene has a beginning and an end. And so this is kind of the typical stop sign that should occur at the end of the gene occurs somewhere in the middle. And the body just says, oh, okay, I guess that's the stop sign, and it stops making the protein, and then you get too short of a version that can't carry out its job, and the body usually kind of decays that. There are insertions where you can add a small piece of DNA. That can be a single letter, that can be 5 letters, that can be 75 letters. It can be a whole extra piece of chromosome that gets added. There are different sizes. And then a deletion removes a letter or a piece of DNA. A duplication can occur where a piece of DNA is copied, and we'll talk a little bit about how that occurs. And then there's something else called the reading frame. So again, these words are 3 letters. And if I stick an extra letter in there, then that kind of shifts everything over by 1 letter and changes all of the words afterwards. And most of the time, that will, at some point in time, turn one of those words into a stop sign. And that will end up causing the protein to stop being formed. And this, again, is called a frame shift mutation. Okay, so I put them in pictures, and so I tried to add a little bit. So the first one is a silent mutation. I don't have a pointer on my thing, but you can see there the first sequence is serine, valine, proline, and it's the A's and then a couple letters there, and then GTA, and that second one is GTT. So we've changed an A to a T at that 6th position. But GTT still codes for valine, so nothing really happened because of that redundancy, and that's called a silent missense mutation. The next one down, you can see that a little bit after that, we changed proline CCC to ACC, and that changed the proline to a threonine. And so that would be a true missense mutation. We substituted one amino acid, one building block of that protein for an entirely different building block that maybe has different properties, maybe it's bigger, it doesn't kind of fold the same way, and so that's a missense variant. The next one at the bottom is a nonsense mutation, right? So you can see that last one is TAC, and instead it's TAG, and that's a stop sign for the protein to say, oops, I guess this is the end. We're done making things, even though they shouldn't have been. And then the frame shift mutation, again, you can see at the top, there was, they inserted a couple of C's there at the beginning. Instead of GTA, you got GCCC, and then it kind of shifted everything, and we've got different amino acids there, okay? And then the last one is an example of a duplication. So this is kind of a picture of chromosomes. You may also have heard the term arms of chromosomes, right? So this is kind of depicting those arms. The one at the top is the short arm or the P arm, and then the one at the bottom is the Q arm or the long arm. And the green portion, they've copied twice onto that long arm. So you get an extra portion of DNA, and maybe that extra portion contained another GRIN2B gene. Okay, so that was kind of the basic information about the types of variations. This is what might show up on a genetic report that could help you interpret what kind of variation is present. So you'll see up there the first letter, C, stands for coding. That tells you the address of the variant in the DNA. The P next to it is the protein address, so that tells you in the final product of the GRIN2B protein, where is this located? And you'll see that 1345 divided by 3 is 448, right? Because that's our words, they are 3 letters. And so those each are divisible, the bigger one is divisible by 3 to get you the second one. And then the top part there tells you, okay, at position 1345 in the coding sequence, we were supposed to have an A, and it became a G. And in the protein, that means that at position 448, there was supposed to be a phenylalanine, and now there's a tyrosine. And these abbreviations for the amino acids, you can look them up online. They have both 3-letter abbreviations and 1-letter abbreviations. We also talked a little bit about the arms in the previous slide. Okay, so the next thing to know is that most of the time variations in GRIN2B are all called heterozygous. Again, we have 2 copies of every chromosome, one from mom, one from dad. Being heterozygous means that one of those copies is not working. And for GRIN2B, that's enough to cause the symptoms of the disease. There are other conditions where you have to have both copies not working. Those are called recessive. There are also ones that are formed on our X and Y chromosomes that have different names, but for our purposes, GRIN2B is autosomal dominant. Autosome meaning our non-sex chromosomes, dominant meaning one affected copy is enough to cause symptoms. Most of these variations occur just in the parent or just in the patient. So, we're talking about it as a genetic disease, but a genetic disease does not always mean an inherited disease. It just means, hey, this has to do with your genes. The genes are things in our body that give us the instructions to make our bones, to make our brain, to make our kidneys. And so it's an alteration in one of those genes. Sometimes there can be an inherited variant, but usually that means that the parent is a mosaic, meaning that some of the cells in their body contain the variation and some of the cells do not. And I think that's been maybe reported in 1 or 2 GRIN2B patients. It's much more common in GRIN2A, but it has been reported. So most of the time when we do this type of testing, we recommend that families get tested as well to make sure that, that the variant is not inherited from a parent who has only some cells affected. The risk in that situation is for recurrence is very, very low. Obviously, we didn't test sperm cells or egg cells, so we can't rule out that the variation exists there, but the risk is very low. And that's what I'm talking about here in this slide, right? So mosaicism is when there are some cells in the body that have the variation and some cells that do not. And usually it happens from something like this. The variation occurs when the genome is being copied over at some point in cell division when the baby is growing, and then all the cells descended from that particular cell will carry the variation, whereas many cells in the body will not. And so that is called somatic mosaicism, meaning our body has the mosaic, but germline mosaicism can still exist. So cells in the egg and sperm cells can still have variation that we don't test. Okay. So the next thing to talk about is like, well, how did this all happen? Why does this variation happen? And again, usually it is when the cells are being copied and cell division is occurring in the early fetal period, but it's typically not due to anything in particular. It's not the piece of rotten cheese that you ate from the refrigerator, or the teeny car accident, or the medicine that people took before they knew they were pregnant. It's usually not not due to any of those things. It is the fact that we all contain genetic variation. Our machinery is not 100% accurate in copying over all of our genetics. And it's designed to be that way. It's not supposed to be 100% accurate because then we would never have any evolution. So, I think this is just saying that that variation happened in a spot where it was really important to have the letter that was supposed to be there. Okay, so this I put in as an example of some test results, and just showing again that it will tell you what type of specimen. So I think it's okay to have testing for these types of conditions from blood or saliva. Overall interpretation, so usually they say positive, meaning that they found something, not positive like this is great. It's just meaning that the test showed something. And then they will give you an overall summary and then the specific results then at the bottom. And so usually they will show, again, first the gene name. They will give you the address. They will say, again, whether it's present on one or both copies of the gene. Those are called alleles. And then last, they will classify it. So we talked about, pathogenic versus benign versus a variant of uncertain significance. Most of the time, people group together likely pathogenic and pathogenic as probably, the answer for things as the disease-causing result. Okay, that was all the genetics. Okay. Last section, clinical presentation. Okay, so this is also AI-generated summary of things. These are kind of some of the features that we see in GRIN2B, and this came from I think 133 cases that have been published so far. So obviously there are more bits of information out there that are not in the medical literature, but this is kind of an overall summary of some of the cases. So I think doesn't need to be said here, but the most common feature being neurodevelopmental issues and intellectual disability, but also very common being hypotonia, the abnormal brain or MRI we talked about with a malformation in cortical development in double digits of people with GRIN2B. And then epilepsy also being quite common with a number of different types of syndromes. The other features that have been seen include language issues, autism, a small head size, and then the malformations that we talked about. And then finally, these things I think are not that they are less frequent necessarily, but also not always reported in a number of the different papers. And so they don't have as good of a quantity for them, but sleep difficulties, movement disorders, and CVI. So, I'm sorry, it was 105 patients. I thought it was 130, but 105. They also did another study, I think this was by Dr. Simons in the UK, that if you looked at all, based on their national health data, if you looked at all the diagnosed neurodevelopmental disorders with epilepsy, the prevalence was about 0.2% for GRIN2B as one of those diagnoses. And of those 105 patients, 84 variants have been published. So that means that most people have a unique variation that causes the GRIN2B diagnosis. And those ones that are associated with how the brain forms tend to cluster in a couple of places. So I put them there, and I put that drawing again for our reference. So the M3 domain, the S2 domain, and then between S2 and S4. And this is very similar to GRIN1, where malformations of cortical development are also seen. And then the gain of function variants typically cluster in a couple of places as well. There are a few recurrent variants there at those particular spots, 820 and 689. But again, most of them are unique to each person. Epilepsy, as we said, is kind of common, and the age of onset was from infancy all the way up to 9 years of age. A lot of kids had different types of seizures. Some kids just maybe had a few, while others, had them quite often, and about half were treatment resistant. I know there's going to be another talk about this, but I just wanted to throw it out there because, again, this is supposed to be kind of the basics. So a lot of people will talk about this as, kind of what does this variation do to the GRIN protein? Is it a gain of function or a loss of function? So basically, when the protein is not working well, that's also referred to as haploinsufficiency. Again, one copy not working, half not working. And then gain of function means that the protein has acquired some different properties. Maybe it's a bit more sensitive, maybe it binds something tighter than it should. The analogy that I heard was like, if you put a different cog kind of in the car as you're putting it through the assembly line, most of the time the engine may not work. But if you put in a certain different type of part, then perhaps the engine will go faster, or it will accelerate more quickly, or will use less gas, something that is extra that wasn't the purpose of what you were trying to do. And sometimes this gain or loss of function can be inferred from the type of variation. So if you stop the protein too early, if you delete the whole protein, if you shift everything and you get a stop sign down the road, those are all going to result in loss of function. So it's really the missense variants that are the ones that are more tough to figure out. Those ones where just one letter is substituted for a different letter. And that's pretty tricky. And that's why we have, all these people working on it, right? So that's why we can do functional testing. We're trying to work on algorithms to help determine which types of variants are more likely to be one or the other. And again, sometimes there are hotspots for different types of variations that, again, can help us to understand that. Okay, the next section I put in was not on the original outline. I think I'm still doing okay, but this is about how to get to the portal and how to navigate the portal. Thank you. Going there is kind of the quickest way to understand what is going on with a family member's variant. So the top green bar there has the section for variant classification. You want to enter GRIN2B from the drop-down menu because it's all NMDA receptors. And so you want GRIN2B. You have to clear all of the stuff that's already there. And then enter the particular variant that's on the report. There are sometimes, I've talked to a lot of families that have trouble and they kind of they're like, well, it didn't work, And sometimes that is because if you have an older test report, we didn't know exactly where all the genes started and stopped. And so we kept kind of changing things, and we have these called transcripts. And so if there's an older transcript, it may not translate correctly. And in that situation, I recommend using the P coordinates rather than the C coordinates because it may very well work if you do it that way. So this is the portal. And again, I've entered in a variant. I entered in a GRIN2B variant. And what I see there is the categorization, right? It tells me it's likely pathogenic. It tells me the functional information on the right if I scroll down a little bit, and I can see that the analysis has been completed and that this is likely a loss of function variant. And then I can also compare it to other things. And so this compares it to other variants in the region of the protein that is similar to the region where this variant is located. And it tells me that there is one other patient that has this same variant in the database. Okay, so I think this is my last slide. So these were some references. There's kind of some science-y papers there at the top, and then some links to a number of various places that have really good, accurate information about the GRIN2B diagnosis, about genetics, about clinical symptoms, all sorts of things. Ta-da! All right. Phew. did a good job. Everyone understands all of genetics, so we're good.
So, for purposes of the live stream, if you do have a question, I'm going to move this microphone to you so that you can articulate it so that people on YouTube will be able to hear the question. So.
Well, and I'm happy to talk to people afterwards if they don't want everyone to hear their question. I'm okay with that too.
We can wait another few seconds. Sometimes it takes time for people to formulate questions in their mind. Totally.
Hi, Kristen.
Hi.
Hi. If I see that we know from so many mutations, basically only the missins, we don't know if it's a gain or a loss of function. Why isn't, in this case then, loss of function a standard item in the genetic reports? Because there are so many parents waiting for that and being held up.
Yeah. Well, I think that that is kind of a terminology that is not necessarily part of what the genetic analysis is supposed to give as an answer. And so I think they will probably not take that extra step to do that. I think that's the role of us as the GRIN community to kind of communicate that, because it can be different for different genes. It can, kind of carry different weight in different situations. And so I think the genetic companies are not going to include that as part of their standard process. They're very regulated in what they put on the report. They have to go through this big kind of process to give you that pathogenic or uncertain or benign classification, and it doesn't say anything about function. So I think that's our job to kind of get out there.
While I hold a little bit longer for additional questions, I just want to remind everybody you have a QR code on your table, so if you're more comfortable putting something in that we can get back to during some other Q&A panels, you're certainly welcome to use that as well. Okay, one more time for Dr. Park. Thank you. Okay, we're actually ahead of schedule by, I think, about 9 minutes. So at 3:55, our next presenter will be coming. And then after that, we have a parent presentation. And then after that, cocktail hour. And also, our bee mascot will be present for photos. And Spider-Man. Yeah. You're right. Dr. Park, you probably remember we couldn't get Godzilla Festival here this year. That also happened, I think, was it 4 years ago? In Chicago. Yeah, Chicago. Shout out to my town. Godzilla Festival was happening in the same hotel as our conference, and a number of scientists and researchers may or may not have partied down with the Godzilla people. I've seen pictures. It's pretty cool. All right, yeah, so then we'll pause for just a second, but yeah, right around 3:55, our next presenter will be here. All right, everybody. We're going to move on to our next speaker. And always a pleasure to introduce to you all Dr. Tim Benke. He is he's an MD at the University of Colorado. And he's here to speak, kind of piggybacking off of Dr. Park's Beginner's Guide to GRIN2B, and sort of this is now the next step in the process, which is understanding loss of function versus gain of function variants. So please welcome Dr. Tim Benke.
Am I in control of the slides? Oh, there it is. Hi, everybody. So this is what happens when you go to family meetings. It's what you bring home. So just be careful, take care of yourselves, and don't bring this home. I had this 2 weeks ago. It's from the RET meeting, and it's just, I'm not infectious, but it's still hanging on. So I want to talk to you about gain of function and loss of function variants. And really, this is meant to be directed at you because you're asking the question, what is this, and what do I do with this information? Everybody keeps talking about it, and I don't know what to do with this information. So these are my disclosures. So I want to review some genetic concepts and terminology, discuss the genetics of GRIN disorders, and then understand how to interpret a genetic testing report, understand what we mean by functional analyses, and understand the limitations and utility of functional testing. So, clinical testing, you know how this happened. So based on signs and symptoms, your clinicians ordered a genetic test. And there are a lot of different genetic tests, each with their own limitations, and that's a longer lecture, but it might have been a chromosomal microarray where it looks for large chunks of DNA that are missing, duplicated, or rearranged, or a genetic panel where the coding sequence of a small set of genes known to cause a particular disease were analyzed, or it could have been whole exome sequencing, which analyzes the coding sequences about 20 to 25,000 genes. But it's important to note that this is less than 5% of all our genetic material, and about 5,000 genes are known to be associated with disease. So trio is the best version of this, and it's when the affected child is compared to unaffected parents. Then there's whole genome sequencing, which analyzes the sequence of all of our genetic material. And this is the best, and it compares to unaffected parents. So trio is best as well, compares to unaffected parents. So in several weeks, you know what happens. The report came back, and the report had a lot of information, including an interpretation. And your clinician applies the interpretation to your symptoms to determine whether the test results explains what's going on. So your clinician determines how this then affects your care plan, unless it's more complicated than that. So this is an example test report. And this is, of course, for not a GRIN2B disorder. I couldn't do that because that wouldn't be fair. But basically, it'll say what is the gene involved, MECP2 in this case. It says the coding change. And this is important because that's the address in the DNA for this particular change in that gene. And then it says, okay, well, if you have a change from a C to a T at this particular address, what happens is it turns an arginine into a cysteine at the protein address, which is, again, related to that DNA address. Basically, the relationship is you divide it by 3. It then talks about zygosity. Is it in both? Both, you get 2 copies, one from your mother, one from your father. Is it in both, or is it just in one? It doesn't tell you which one. It just tells you that you either have both or one, which is heterozygous. And then it can be really clear and say, well, this is a pathogenic variant, which means it's disease-causing. In some cases, there's then an interpretation down at the bottom, and the interpretation depends a lot on the company that did the test. In some cases, you will get all sorts of information that may or may not be accurate. And in the case of GRIN disorders, sometimes it even suggests treatments which may not be relevant because this test does not tell you whether or not you're gain or loss of function, unless your particular variant is well characterized before and the company happens to look that up in the literature and put that into your test. So where did this interpretation come from? So the testing lab compares their sequence data to existing normative database, so it's people without the disease. It also compares it to a database of people with similar disease, and these are curated by experts who are familiar with specific diseases and their specific genes. There is a panel associated with GRIN disorders of experts who say, yes, this is disease-causing. No, it is not. So there's a panel for that, for you. And then they have their internal database as well. So the possible results on your report can say things like pathogenic and likely pathogenic, meaning it's disease-causing, benign, not disease-causing, or it's a variant of uncertain significance, which is uncertain. We don't know. We don't want to weigh one way or another on this particular case. So just to take a step back, how is our genetic material doing what it's doing? Well, every cell in the body has inside the cell, in the nucleus, a central part of the cell, is the genetic material, and it's arranged into chromosomes. And those chromosomes are basically DNA that's transport materials and send signals, protect the body from viruses and bacteria, carry out chemical reactions. And basically, the recipe needs to go to what you're trying to get, which is hopefully a nice, beautiful chocolate cake. And that's the protein. And the proteins are made up of smaller units called amino acids. There's 20 different types of amino acids, and the sequence of amino acids determines each protein's three-dimensional structure and specific function. It's like your recipe. You've got to put them in a specific order in order to get what you want to get in terms of the cake you want. So those are the ingredients that go into your cake. So exons and introns. So the DNA material is not one continuous recipe. There are commercials between each one of the chapters. And you can imagine that you could do without the commercials between the chapters if you just linked it all together. Well, basically what happens is you start with DNA that's got the interesting bit, the chapters, and you've got the commercials between the chapters. Well, what happens is when you convert the DNA to the RNA, you still have those commercials in there. But then eventually, all of those commercials get spliced out, and then you finally get the messenger RNA that's ready to make your protein. So an exon, that's the segment of the DNA or RNA containing information coding for a protein. It's the expressed region. It's the chapters. Whereas the intron, it's the commercials. It's the non-coding region of DNA that is removed before a protein is made. And intron stands for intervening or intervening region. It does have, even though it's a commercial, there could still be some important information in there, like containing regulatory elements such as enhancers. And it's also important is that these introns serve as a purpose because you can alternatively splice how you put those different chapters or exons into the sequence that you want. It gets really complicated about how beautifully we are made in order to do these different things. What is a genetic variant? So a variant is any alteration in the DNA sequence that's different from what is seen in the majority of people. And a variant can be benign, like I said, pathogenic, or of uncertain significance. And a pathogenic disease-causing variants are referred to as mutations, and we sometimes go back and forth between those different terminology. So the types of mutations that you can have are a missense mutation. So this is a change in one DNA base pair that results in the substitution of one amino acid for another. And always remember that these are read in 3 DNA letter chunks. So a silent mutation changes the DNA, but not the resulting amino acid. And this is because it turns out that code, the 3-letter code, is somewhat redundant. A non-sense mutation is a change in one DNA base pair that signals the cell to stop building the protein too early, causing a shortened protein or early truncation. Now, there are rules, and I put quotes around the rules. They're air quotes because there's really guidelines as to when or when this may not happen, resulting in nonsense mediated decay. So in other words, the code says stop here. It doesn't mean you end up making half of a protein, but there are rules to say, okay, I might make part of the protein, but the body recognizes that as, oh, there's been a mistake here. We're going to actually make the RNA go away, nonsense mediated decay. Or it may decide, based on these rules, we will make a chunk of the protein. An insertion adds a piece of DNA. A deletion removes a piece of DNA. A duplication is a piece of DNA that's copied one or more times. So if a deletion, insertion, or duplication alters the reading frame, that is that 3 DNA letter code gets out of sync, then this often results in an early truncation. So here's some very simplistic ways of trying to think about it, but I still think it's important. So if the normal recipe says the cat ate the rat, a little substitution turns into the car ate the rat, which may not end up with the sort of recipe that you were looking for. A nonsense mutation, the cat ate the rat, turns into just the cat. So an incomplete story. An in-frame insertion, you go from the cat ate the rat to the bat cat ate the rat. So it's in frame, but you still may end up with something that's completely outside of the sort of thing that you were trying to communicate. An in-frame deletion or duplication happens, the cat ate the rat, and now it goes to the cat ate rat or the cat ate the rat. So again, you're ending up with something that may be in frame, it may still convey the information that you wanted, or more likely it may not. Frame shift mutations, these are out-of-frame deletions and insertions, so basically now you've got something that you've put an extra C, or you've lost a letter here, and that three-letter code now gets completely disrupted, and now you've got a bunch of gobbledygook. So instead of the cat ate the rat, you've got the cat act ev era t. And anyway, what happens is eventually you end up with a stop, but you may have some gobbledygook before you end up with the stop. So this is what often happens. We have a missense change. It's classified as a variant of uncertain significance. Now what and why? Your clinicians ultimately decide, but they reach out for help. And so they're going to ask the question, they're going to ask us or anybody else what's been reported in the literature, and they're going to reach out to the registry, which we'll talk about in a second, for help. So one of the important things to think about is what part of the protein is affected? Is it a domain that we understand well enough to check the function? Well, if there's a problem in one of the introns, it turns out that we don't have necessarily good functional tests to say that that intron, how it's affected, is going to affect the overall function of how things get assembled together. So the exons, the protein sequence, somehow folds together to form a subunit. And so basically, I'm not sure if my pointer works. No, it doesn't. But basically, if you look at the linear sequence of how a GRIN2B subunit is encoded, it goes from the A-terminal domain to S1 to then these different ligand binding domains that are interspersed throughout, finally ends up with a C-terminal domain. And that whole sequence gets folded into this cartoon that you can see how the linear strands kind of go in and out after the whole protein is folded to form a GRIN2B subunit. And then it's important to note at the end of all of this, you've got this long squiggly thing called the C-terminal domain, which does a lot of important things that we're only beginning to understand, but in the case of the GRIN2B is very important as to how NMDA receptors function. So the overall hypothesis is that any sort of change is going to affect the function, and this is going to affect the phenotypes, the symptoms that you're seeing, and potentially treatments. And this is the long list that many of you are very well familiar. Developmental milestones, regression, epilepsy, storms, changes in your MRI or EEG, vision, movement disorders, etc. So what is 5 minutes? Oh, geez. So what is functional analyses? So mutations are engineered into the DNA sequence coding for the receptor, no introns, and that's important. We're completely skipping that important feature there. These are placed into cells that normally do not express the NMDA receptors, and the properties of NMDA receptors are then studied, and these are compared to unmutated receptors. And we're asking a lot of questions about how these receptors function. Dr. Train Ellis gave a talk today. This whole notion of gain of function or loss of function, it's really outdated, but it's what we have to work with. It's a lot more complicated than just gain or loss. So do the receptors bind glutamate, that's the main neurotransmitter, more or less? Do the receptors bind glycine? It's a co-transmitter, more or less? Do these receptors get blocked by magnesium and other regulatory ions? Do these receptors open and close, more or less? And when they do open or close, do they stay open longer or less? And do these receptors actually make it to the surface of their cell, or do they get somehow gummed up in the machinery trying to get there? And once they get to the surface, do these receptors bind to who they are supposed to bind to via the C-terminal domain? So basically, what happened is we looked at all of these different properties, and we assembled a point system to decide if overall this results in a gain or loss of function. And it's, of course, divided into likely gain, possible gain, indeterminate, possible loss, likely loss. So it's a spectrum. And what often happens is you end up with a mutated receptor that's got a few points on this side of gain and a few points on this side of loss. We know that it's not working correctly, but the points cancel each other out and you end up with indeterminate. So there are a lot of limitations to functional testing. Are we testing for everything? We're testing for what we can. Do neurons behave the same way as these cells that we're using for these tests? Probably not. Does it depend on the type of neuron? Oh yes, very much so. Should this be studied in whole animals, neurons derived from humans? And why don't we just get AI to figure all of this out? So one of the things that we know from prior studies, if we look at GRIN2B, that there's a spectrum of different things when you look at all of the different mutations and lump them together. Now, in this older study, it's important to note this was an older study where intellectual disability was isolated in this particular case. And so almost everybody with GRIN2B has some degree of either developmental delay or intellectual disability. While this says 55%, when you add in developmental disability, it's probably going to be closer to 100. And that's why we're now moving to, within the registry, from not just studying a few patients, but hundreds of patients. But it turns out that the C-terminal domain, we now have a lot more variance in the C-terminal domain, where we found that the truncating variants, the nonsense variants, there's 100% intellectual disability. There's a lot more autism spectrum disorder seen in the truncating variants. Whereas epilepsy, it's about the same rate as is seen in overall in the other variants. And we hope to be submitting, basically, we've come up with a new assay to try to assess this. So this is the question that you're often asking. Where is my functional analysis? And you often email me, where's my functional analysis? So what happens is full enrollment in the registry is required, and families contact us at grinresearch@cuanschutz.edu. Kiara, where are you? Or are you out in the hallway? There she is, Kiara, please stand up. So you can maybe bypass a lot of that if you see Kiara today or tomorrow. So what you have to do is families email copies of the genetic report, all the pages. Myself and Dr. Park, we verify whether or not you're eligible for enrollment. You work with Kiara to get the online consents done. You then complete the questionnaire, and then we notify Dr. Trinellis in his lab at Emory to proceed with the functional analysis. So the functional analysis is provided to your physician at their request, but they can contact us, or they can look it up on the portal. The Trinellis lab uploads the reports to the portal. We try not to provide these directly to the families, because it implies on our part that we have a treating relationship with you. No, we don't. We have a research relationship with you. I'm happy to talk to anybody, but I have to be careful to speak to you in very generic terms. When a variant comes into the Trinellis lab, it's important to note that it takes a while for these to be processed, and they're prioritized based on a lot of different factors. But the bottom line is it takes several months. It goes to a very large factory. There's lots of smokestacks in that factory. I'm a wholly owned subsidiary where we test the C-terminal variants. And I should have told ChatGPT to put a really rusty bicycle in front. So this is viewing the registry. You go to the portal. If you want to enroll in the registry, if you're in this half of the world, you email us. What do I do with my functional analyses? So, like I said, these are not clinically actionable. We're using them to how you get enrolled into a research study or natural history studies. Your clinician may decide to use them for something else, but that is up to your clinician to do. But one of the examples is this, is that future clinical trials may include only gain of function or loss of function. And while I say future, we are in the future right now because we know that that's happening for the Radipril trial. So why? The reason is that if you are a gain of function, you probably don't want to be on a drug that is going to cause gain of function or vice versa. You don't want to add insult to injury is the bottom line. This is what the portal looks like. I'm just quickly going over these because I'm at time. And what I really want to do is to thank you, my lab, my partners, and the funders, which include you guys, GRIN2B. Hey, I did that pretty good. I got right to when she held up the sign. And my voice still hung in there, barely.
We have a couple of minutes for questions for Dr. Benke, if anybody has anything. Okay.
For those variants that have tested indeterminate, are there plans to retest or test another way?
So the question is if your functional report comes back as indeterminate, what else are you going to do for me? Well, at the moment, it is what it is. But the idea is that those receptors are not functioning correctly. You've got points on either side, and it's likely that those folks are going to be included in future clinical trials. So at the moment, the answer is no. Anything's possible in the future because let's say an indeterminate receptor is engineered in a different way into a new system. And we heard about this today. One of the grants funded by GRIN2B is to try to find new ways of testing the indeterminate ones. We may find that, oh, no, you're actually a gain. So, but I think it's still on the horizon. Dr. Penn and the University of Sussex is really trying to crank this up to get that going to answer that question better.
So if there are multiple GRIN2B people with the same variant and they're already in the registry, are they, I guess I have a two-part, do you track who's in the registry? Like, how do I know that Hunter was tested? Or does it matter? Or do you test
it's huge.
multiple times?
I want as many, and this is what's important. So for one of the truncating variants, it turns out it's recurrent. So we see it in a lot of people. I think for the 847X, it's about 15 different folks with that. We want to have as many possible in all the different variants. If you don't know if you're registered or not, you email us and you say, I can't remember, we'll tell you. But if you're not, and we already have your variant, it is really important to us to still get you enrolled. Now, we may not have the carrot and stick for you. The carrot being that if you enroll, you get functional testing, and we may already have functional testing for you. But we still want you to register and fill out our questionnaire because it's important to know that if everybody has this variant, they all look the same or not. How'd I do?
Great.
Thanks. Next question. Oh, there's another one. Somebody raised their hand and got shy.
No, my question was similar about like if we've already gone through this, is there ever a time that we would redo this or like?
So there's a lot of folks that were registered early, and we've gone back to try to reconsent folks because if you, in the before times before the pandemic, we might have only gotten, version 1 of the questionnaire. And when we're now on, Dr. Park, what are we on, like version 15B? And we're now asking for people to do it on an annual basis as well. So if it's been a while, reach back out to us, because maybe we want to get more information from you.
Thank you, Dr. Benke. We're going to let him save his voice. He's got more presentations. Thank you. Okay, so before we get to our final presentation, just a reminder, beginning at 5:30 until 6:30, it's going to be a cocktail hour out in the hallway, and that cocktail hour will feature character photo opportunities featuring the GRIN2B and Spider-Man. Why not? We're pretty sure that one will not eat the other. And then at 6:30, we would want you to be our guest for a dinner and dance party that'll be happening at 6:30 as well. All right. Very excited to hear our last presentation from two queen bees. GRIN2B caregivers, Kelsey and Allison, are going to share their journeys as moms of their kids. So I'm going to invite both of them to come on up. Give them a round of applause for Kelsey Budisa and Allison Dreyer. And then two microphones.
See whose slides go.
Are you going to go first?
I was gonna see whose slides came up first. Okay, I'll go first. Yeah. Well, as he's pulling those up, I just want to say hello to everyone, and I'm excited to be here. My name is Allison Dreyer, and I'm honored to be able to share our story with you. I will say I'm not a public speaker by any means, but I'm hoping that through sharing our family story, I can offer solidarity and resources to people here. So this story is about our 7-year-old bee, Gavin. This is our family. Garrett and I were married in July of 2016. Our two boys are Gavin, who's 7, and Ezra, who's 5. Garrett works full-time as a field technician for an environmental sampling lab, and I'm an occupational therapist for the Winston-Salem Forsyth County School District in North Carolina, where we live. This is Gavin. To start at the very beginning, I wanted to briefly share a couple of things. My pregnancy with Gavin was pretty typical until about 32 weeks when I was diagnosed with intrahepatic cholestasis of pregnancy, or ICP, which is a liver condition that occurs during pregnancy. Because of this, I was induced at 37 weeks, and Gavin was born about 48 hours later. I don't know that ICP is related to GRIN2B, but if it helps with future research, I wanted to share it today. Also, at 9 days old, Gavin was hospitalized for hypothermia and underwent a full sepsis workup, which was negative, thankfully. And that is Gavin's only hospitalization to date, knock on wood. This is our timeline, our diagnosis journey for Gavin. The first few months of Gavin's life, I'm a pediatric occupational therapist, so I'm familiar with developmental milestones and timeframes. So I noticed early on that Gavin wasn't progressing as expected. I brought up my concerns during well-child visits and was just told, let's give it more time, he'll catch up. But around his first birthday, I really pushed for a referral to early intervention because I was especially concerned that he wasn't anywhere near walking. So we were definitely concerned with his gross motor development. Like so many families in 2020, our plans changed when COVID hit. Early intervention services were postponed, and after waiting a couple of months for services to reopen, we decided to pursue outpatient physical therapy on our own instead. In September of 2020, Ezra was born. As Ezra grew, we watched him reach milestone after milestone with what seemed like very little effort. At the same time, every new skill was something Gavin had to work incredibly hard to achieve. Watching the two of them develop side by side made it even clearer that something more was going on. We are now seeing the significance of Gavin's speech, gross motor, and fine motor delays and began searching for our bigger why. We met with a developmental pediatrician expecting that Gavin might be diagnosed with autism. Instead, he was diagnosed with global developmental delay, and further testing was recommended. Basic genetic testing came back negative, and then we were referred to genetics, at which time we found out it was going to be a 15-month wait. In December of 2023, we finally met with the genetic team and had the whole exome sequencing testing done. And then on Valentine's Day of 2024, we received the answer we'd been seeking. It was GRIN2B related development, delays in motor skills, and a moderate cognitive impairment. But despite that, he is mobile and communicates with a mix of words, phrases, sign language, and an AAC device. But beyond our diagnosis of GRIN2B, this is the real Gavin. He's a huge Curious George fan. He loves music. He's the definition of a sensory seeker who especially loves water. He's very silly, loves laughing at random noises, especially people sneezing. He also has very strong connections with the people in his daily life. One thing we learned is that the therapies, equipment, and resources available to rare disease families make an incredible difference. Many of the things we have in our team right now are things we discovered through other families. So I wanted to share what's been helpful for us in case it's helpful to anyone else here today. I'm not going to talk about all of these, but I'm happy to talk about them more if you need more information on any of them. Some things I will point out is under therapies, we have parent-child interaction therapy. This is a form of behavior therapy. We were interested in pursuing ABA therapy for Gavin, but because he doesn't have an autism diagnosis, that wasn't available to us. This was kind of the alternative for that, and it was really beneficial to teach us as parents how to better interact with him on his challenging behaviors. In the equipment box, I've listed the things that Gavin uses to access his world safely. He has an AAC device for communication, an adapted stroller, adapted bike, an easy-on harness for the car, and a safety bed. He also previously had AFOs. The primary medications he uses are L-Serine, which he's been on for about 2 years, and lactulose for chronic constipation. A big part of this is as we all know, is funding for all of these therapy and equipment needs. A little over a year ago, Gavin was finally approved for disability-based Medicaid, known as the CAP-C waiver in North Carolina. Some of the things that CAP-C provides Gavin with are paid respite care and funding for adaptive equipment and activities. Additional ways that we funded some of these things before having Medicaid were through GRIN2B's Patient Assistant Grant, and also through Connect, which is an online resource that helps families find grants that they might qualify for. We've also been able to try a variety of adapted activities, which has been really fun, and I've got some of the pictures here. The first two pictures are from Victory Junction Family Weekend, where Gavin got to try horseback riding and bowling. We also have a picture of him trying out adapted softball, which clearly he was not a pro at. And then at the bottom, we have pictures of him with his AAC device and his adapted bike. So what are our biggest challenges today? Chronic constipation has been one of Gavin's biggest medical challenges since toddlerhood. He requires medication management for successful bowel movements, and because of previous trauma with constipation, he's often fearful and avoidant of toileting, especially in public places or with new people. We're thankful that Gavin is daytime potty trained, but he continues to need help with the steps of toileting and needs familiar people and environments to be successful. Behavior is another area we work on continuously. Gavin becomes very overwhelmed with sensory experiences like haircuts or dental visits. He goes into fight or flight mode and becomes physically aggressive during these visits. He's also a wanderer and doesn't yet recognize danger or safety risks. He's walked out of our front door alone multiple times, and just a few months ago, he left a house we were visiting in Charlotte where he made it down the street before we found him. So we use a variety of safety measures, including air tags, safety locks and door alarms at home, and then a variety of doorknob covers and locks when we travel. What does the future look like for our family? For me, it comes down to 2 words, fear and hope. The fear is very real. Every day we think about Gavin's safety. We also wonder what level of independence he'll have as an adult and who will care for him when we're no longer able to. But hope is just as real. Hope is watching Gavin continue to make progress, even if it's at his own pace. Hope is finding communities through the GRIN2B Foundation and Cure GRIN Foundations. And hope is being in this room with all of you, especially knowing that researchers are dedicating their time and talent to developing treatments that could change Gavin's life. So lastly, I just wanted to say thank you to the GRIN2B Foundation, the researchers, clinicians, and families who are here and making this gathering possible. I want to thank you all for reminding us that our children's futures matter and for giving families like ours hope.
Thank you, Allison. Okay. Oh, all right. Okay, first I want to thank Liz, Alyssa, and everyone at the GRIN2B Foundation for organizing such a wonderful and important weekend. And to all of the researchers, doctors, and professionals who are here, I think I can speak for all of the GRIN2B families when I say that we greatly appreciate all of your hard work and dedication to understanding this rare disease. Your work gives our families something incredibly valuable, which is hope for our children's future, and we're grateful that you've chosen to spend your time with us this weekend. My name is Kelsey Bethesda, and I'm honored to be here today to share my experience with GRIN2B thus far. Before I get into the nitty-gritty, I wanted to tell you a little bit about our family, and most importantly, about my best friend, who just so happens to be my 4-year-old son, River. So my husband Dan and I are originally from northern New Jersey, but moved to Maryland in 2015. He's a Navy veteran who works in the intelligence field, and I have a background in the fashion industry. We had our son River in 2022, which is when I became a stay-at-home mom, and most recently we had a baby boy named August, who is 8 months old. This is River. He's my hero. He's the strongest, most resilient little boy in the entire world, and he's been through more in his 4 years of life than most adults ever will. Anyone who meets him instantly falls in love with him. His smile, his big, beautiful eyes, his sense of humor are just a few of the things I love about him. He loves Elmo. He loves Mickey Mouse and Bluey. He loves riding his adaptive bike. He loves rolling his eyes at me when I ask him to do something he doesn't want to do. And he loves being bounced around and having his hands clapped. So the beginning, which was definitely the biggest struggle of our lives. It was my first pregnancy. We were over the moon excited. Everything was going completely normal. All tests were normal until our 20-week anatomy scan. They found enlarged ventricles in his brain and also partial agenesis of the corpus callosum. At 32 weeks, I had a fetal MRI. Before that, everything was going fine. The enlarged ventricles weren't growing anymore. So we were hoping that it would just be, a minor developmental delay or something similar to that. But unfortunately, the 32-week fetal MRI revealed lissencephaly, which for anyone who doesn't know, it's when the ridges of the brain are a little bit smoother. So essentially, the brain has less room to communicate with itself. Our care team wanted us to be induced and give birth at University of Maryland Medical Center, which has a level 4 NICU. And I, instead of being induced, went into spontaneous labor at 38 weeks. I had a very textbook birth, very high APGAR scores when he was born, but he was taken to the NICU immediately for observation, and that was for the best because later that day he started having seizures. On day 4, he got necrolizing enterocolitis, which is very unusual for a full-term baby. It's usually common in preemies. So he had a partial colon and bowel resection, a temporary ostomy bag, and his total time in the NICU was 122 very hard days. And then shortly after discharge, we did receive the GRIN2B diagnosis, which was actually a relief for us because we finally had answers. I was obviously worrying, did that
mozzarella cheese I ate when I was pregnant caused this. I was always worried that it was something I did wrong, but I was glad to know that it was not, and it just happened on its own. Seizures, these are our biggest struggle. In 4 years, River has been through over 10 medications. I'm the type of mom where if a medication isn't making a big difference within a couple months, it's time to wean, it's time to try something else. I don't need him on unnecessary medications. So right now, our current regimen is Xcopri and Briviact. Briviact is kind of like a cousin of Keppra. Xcopri is one of the newer drugs. He's been on it for about a year, and we've actually seen the most success with the least amount of side effects with this one. I highly recommend asking your neurologist about Xcopri if your child is kind of similar to River, if you see any similarities. He does have tonic and focal seizures. They last about 30 seconds to 1 minute. He used to have over 10 per day, but right now he only has about 1, maybe 2 a day. Additional diagnoses, or what I like to say, just added to the list. So he has lissencephaly, which I mentioned, cerebral palsy, laryngomalacia. So if you've met River, you probably instantly notice that he is a noisy breather, and that's his laryngomalacia. It is kind of like just a floppy throat. He did recently have surgery a few months ago. Didn't make a huge improvement, but he kind of uses it as a form of communication. When he's more tired or mad at us, he'll breathe louder, so we can tell. He has severe reflux. He does have a GJ tube, which he is fed through. He has CVI, and then he also has developmental delay. One thing I wanted to mention with our additional diagnoses are things that we're always trying to improve, which is head and trunk control. He has mixed tone, but his head control has always been his biggest issue. With gross motor development, although he's doing really well now with his head control. Standing and walking, he cannot stand independently or walk independently, but he walks in a gait trainer. Fine motor skills we're always working on, so reaching, grabbing, activating toys. Communication we're always working on, so he is nonverbal, but he does use an eye gaze AAC device. Another thing is toileting. He does go on the potty on a schedule we have him on, but because of the lack of communication, he's not potty trained. Tracking an eye contact or something we're always working on and that he's improving with. And then eating by mouth. So he actually ate fully by mouth almost his entire life up until a year ago when he lost his appetite during a clinical trial and had to start being tube fed again. He's now fully fed through the J tube because of his severe reflux, but he enjoys tastes of food, and we're trying to build back those oral skills. Therapy. So these are all the therapies that he has done or does do. Napa Center, we've done a few intensives with them. All kids are perfect. They specialize in DMI therapy, which is incredible. I highly recommend anyone to try DMI therapy if you have a kiddo who has delays in gross motor development. Aqua therapy, hippotherapy, which we love. He loves going on the horse. We have a wonderful therapist who we work with who's seen him since he was a baby. And then, of course, OT and speech. We also have adaptive equipment, lots of it. Our house is like a little supply closet for adaptive equipment, but these are just a few things. I won't go through all of them, but some things I will point out is a Rift and activity chair, which is great for indoors and, moving him around the house and eating and using his eye gaze device. The Freedom Concepts adaptive trike is absolutely wonderful. He has so much fun with it, and we also actually got that through a grant. And then the Tobii Dynavox eye gaze device is the one that he uses for communication, and then also Switch toys, which are very fun. And lastly, this is what I wish every parent knew. So Join Facebook groups. The GRIN2B Parent Support Group is a wealth of information. I have learned more through the advice of other parents on Facebook groups than I probably ever will through Google searches, so I appreciate that so much. Early intervention is key. Get your child into therapy as soon as possible. River started PT and OT through the Maryland Infants and Toddlers program within a month of coming home from the NICU, and it definitely has made a big difference. Research, research, research. Knowledge is power, and it'll make you the best advocate for your child. Research different types of medications, therapies, doctors. Don't be afraid to switch providers if you're not vibing with one. You gotta move on and find your A team, find someone who will support you and really try to make you and your child's life better. Research clinical trials and also utilizing grants for adaptive equipment and therapies. So we have gotten lots of great equipment from the Maryland LIST grant and then also the Fellow Family Fund. Find something for you and take care of your mental and physical health. You are allowed to grieve and be sad once in a while. I'll be the first to say, we've been through a lot. But you can't be the best parent for your child unless you are also taking care of yourself too. For me, I decided to take my knowledge from my career in the fashion and retail industry to create an apparel brand centered around kindness and inclusion. It's called Raise Kind Humans. We're on Instagram. Try to stay organized, schedules, shared calendars, written out medication lists with dosages help me stay sane. There's no other way I could survive. And give your child every opportunity and experience possible. Don't be afraid to travel. Don't be afraid to go to restaurants. Buy the handicap seats at the Yankees game. Get the disability pass at Disney. It may take a little more effort and planning, and you may be exhausted afterwards, but I promise it'll be worth it to give your child those experiences. And lastly, educate your family and friends. I've noticed that the more people know about GRIN2B and specifically about River and his abilities and needs, the more comfortable they become interacting with him directly and understanding him better, which is so important because I never want anyone to be intimidated by him. After all, he's just a 4-year-old kid. And that's all I have. Thank you. I did put my personal email on there. I'm an open book. If anyone ever wants to reach out, ask any questions, I'm happy to help. I'm happy to be a resource for anybody. So thank you so much.
Thanks again, Kelsey. Thank you, Allison, as well. That was awesome. Well, that kind of concludes today's formal agenda of meetings. Thank you, everybody, researchers especially that have been here for the bulk of the day, and families that came this afternoon. Just a reminder, the cocktail hour is going to start at 5:30. It's going to last for an hour, so don't feel like you have to rush down. Also, Spider-Man and the Bee will be available for photo opportunities. And then at 6:30 will be a Bee Hour guest dinner, and everybody is invited, including our bees and other children and whomever else would like to attend that is part of this conference. And then just one more reminder. So tomorrow, starting at 6:45 in Salon 3, which is the room immediately behind us, breakfast will be served 6:45 until 8. Childcare opens in the Chesapeake Annapolis room. That's the room where the researchers were today. So childcare is going to open at 7:30 and go until 5 in the afternoon. And our formal program will begin at 8 a.m. in this room sharp. Thank you, everybody, and we'll see you at some points tonight, I'm sure. Thank you.